have a number of colonies on sample number 2 from one manure area. We have a kind of a different spectrum of colonies on sample number 4, not quite so many, but we see that some of them are a little bit different there, a little bit yellow. Now we're going to replica plate. We want to see whether or not there are any organisms that grew and produced colonies on these plates that will now grow in high concentrations of copper. So what we're going to do is, with each of these series, I've labeled some other plates. We've got one control, which will be the same medium, it's called R2A, with no copper, and then we've got some that have 0.5 millimolar copper. We've got some plates that have 1 millimolar copper, we labeled them on the side, and then this one has 2 millimolar copper, and you can see that it's got a little bit of a bluish tinge due to the copper that we've added. So to do a replica plating, it's very easy. We're going to take our forceps and dip it in alcohol and very briefly get the flame on the end, trying not to get it onto the plastic. And then we're going to take a sterile piece of velvet, simply velvet that you buy at a store. I've already ethanol cleaned this rack here, and I've cleaned this, and we're going to very gently put this over the velvet to make it tight, and I'm trying not to get my fingers on the edge. Now we're going to take this plate, I'm going to label the top of it, I'm going to mark it, and I'm going to mark all of these plates also so that I line them up at the same position. I'm going to try to do them in that order also, because that is my 0.5 plate. And you'll notice that I have a little mark on this. So we're going to line all the plates up so that we'll basically doing a Xerox, stamping the cells from this plate onto all of the other plates. In this way, I'm going to put my plate down, line it up with that mark onto the velvet, press it lightly, and you see that the cells are still there, but it's got a bit of an impression now, and you can actually see them on the velvet. So now we're going to take one of the plates, and I'm simply going to line up that mark with this, press it down and up, and it's going to pick up enough cells so that they will grow and the colonies will be identical in distribution to what was on my original plate. And we can do several little Xeroxes with this, with my high-tech Xerox machine. And then our last one should be identical to this, and this is actually kind of a test to make sure that we didn't rub anything off or otherwise contaminate anything. So everything should grow on this because everything grew on the initial plate. And that's it. It takes just seconds. We are going to take this one off. We'll go ahead and autoclave that. And in the meantime, I will clean this off again on the inside. This is the original plate that we spread from a manure sample. And you can see that there are a lot of different types of colony morphologies on here. And then when we did the replica plating, we took the velvet and then pressed it onto this plate. And then we kind of Xeroxed onto 4 other plates. So this is the plate that had 0.5 millimolar copper in it, and you can see that there are quite a few colonies on there, but they're not as many as on here. So that means that many of the colonies that grew on the original plate could not grow on 0.5 millimolar copper. Now if we go to the next one, this is 1 millimolar copper. You can see there was the 1, and we have even fewer colonies that can grow on this. So those are cells that give rise to colonies on this plate. So, but that means that they're still resistant to 1 millimolar copper. And then here is our plate that had 2 millimolar copper on it. and you can see it's got kind of a bluish green tinge from the copper, and almost nothing grew on that. There might be one little colony up there at the top, but we'll continue incubating to see if it goes. And then if you look at the last plate that we did, this was a plate that had no copper on it, and it should be almost identical to the original one if the Xeroxing worked. And I'll try to line these up properly. And you can see that even though there's a little bit of smearing, most of the things that were on the original plate show up in the same position on this other plate that we did. So that's the replica plating.